Hello, my name is Prashik Hariprasad, and I have prepared a presentation for my assignment for the module BPT 1501 Assessment 6. My UNISA student number is 184028552. Well, in my presentation, I'm going to talk about DNA, the code of life. We're going to look at what is DNA, where is DNA found, the shape of DNA, and what does DNA do. So let's look at what is DNA. DNA is a nucleic acid and is known as deoxyribonucleic acid. DNA is found within every cell of every living thing, and it's found in the nucleus of a cell. So when we talk about that it's found in the nucleus of a cell, it's mostly referred to eukaryotic cells, such as plant and animal cells, where DNA is stored tightly coiled into chromosomes within the nucleus. DNA is also found in the mitochondria, which is known as mtDNA. It is found in very little amounts, and we actually inherit mtDNA from our maternal parents, such as our mother. DNA is also found within the chloroplast of a plant cell, and it's also found outside the nucleus. So, as an example, where it's found outside the nucleus, such as in prokaryotic cells, let's look at a bacteria, where there is no nucleus present. So, DNA floats freely in the cytoplasm, bundled in regions called the nucleoid. Um, so since DNA is found, this type of DNA is found outside the nucleus, it is known as extranuclear DNA. And since also DNA is bundled up tightly coiled into chromosomes, chromosomes work to build proteins during the process of protein synthesis and also assist in the duplication or division process of cells. So when we talk about the duplication process, we refer to um, mitosis, where um, a cell makes a copy of another cell, identical copy. We talk about the division of cells, where we refer to the process of meiosis. Let's look at the shape of DNA. What does DNA look like? So under a microscope, DNA looks like a twisted shape. It looks like a twisted ladder shape known as a double helix shape. Hence, it has two twisted strands. Um, in a non-dividing cell, DNA has long structures called chromatin network, and within a dividing cell, after DNA has replicated, it has short visible chromosomes. Um, Also, a fun fact: within humans, each nucleus contains 46 chromosomes or 23 pairs of chromosomes. Um, since DNA has this unique double helix structure, um, it allows it to be the hereditary molecule, and then it allows it to store instructions for the directing cell activity. So when we talk about that, we basically refer to DNA contains genetic instructions to run living things. It controls all the functions of the cell, controls the function. And DNA also gives us it makes up who we are genetically. Um, that as well. Let's look at DNA. What DNA is made up of. So DNA, as we said, is known as deoxyribonucleic acid, where deoxyribo means sugar and nucleic acid refers to is known as a macromolecule which stores genetic material. So DNA is a sugar which stores genetic material. Um, is a long molecule which is made out of monomers or units called nucleotides. So a nucleotide is basically a building block of DNA. An easy way to think of this is like a Lego piece in a very long chain. Each DNA molecule is made out of millions of these. So each nucleotide has three parts: a phosphate group, a deoxyribose sugar. Hence, it's called deoxyribose sugar because this sugar is only found within DNA. It also has a nitrogenous base. Let's look at what nucleotides more internucleotides. So the backbone of DNA is formed by a sugar phosphate bond. So we refer to this by an alternating a sugar and phosphate bond held together tightly. So where you can see the phosphate joins into a sugar, and then the sugar joins to another phosphate, etc. And nucleotides are joined by weak hydrogen bonds. So the bases meet together from each nucleotide and is held by a weak hydrogen bond. And the rungs of the ladder are formed by four nitrogen bases, which are held together by those weak nitrogenous bonds. So let's look at these nitrogen bases. We have adenine, thymine, cytosine, and guanine. So adenine and guanine are double structured, so hence they are purines. And thymine and cytosine are singular structured bases, hence they call pyrimidines. So let's look at what does DNA look like. Um, as you can see, each base is represented by a letter where adenine is represented as Na, thymine is represented as T, and cytosine is represented by C, and guanine represents G. The base of DNA pairs with each other in a very easy and predictable way, where adenine always pairs with thymine and cytosine always pairs with guanine. Um, they basically join with complementary bases. Complementary bases pair with one another, where adenine pairs with thymine, cytosine pairs with guanine, etc. As the easier way to recognize is the shape, the way it's drawn, the way adenine is drawn, and thymine is drawn. It fits together like a glove. It joins together, so hence it pairs together. So exact same explanation with um, cytosine and guanine. Um, so let's go back to nucleotides, right? So how does nucleotides work, right? So nucleotides um, join together, and this is what. So when we talk about millions of these nucleotides, right? Hence we already spoke about the bases. These nucleotides, as we said, DNA is made out of a double helix structure. This what joins those two strands. So one strand will have a separate nucleotides, where another strand will have another separate nucleotide strand of nucleotides, which is joined together, which is held together by a sugar phosphate bond, each separate strand, and it joins together, right? To um, pair with those bases pair with one another, that causes and it becomes that double helix shape, hence making that DNA structure as we refer to like this, right? So let's look at how does DNA work, right? So DNA stores instructions. So four letters of DNA make up codons. These chemicals are repeated in various orders over and over. These codons make up genes. DNA has short segments called genes as well, right? Um, yeah, DNA also has proteins called histones that wrap around it as well. So when we talk about these codons make up genes, these genes tells the cell how to make a protein and controls everything in the cell. So what how so basically after this they talk about the process of translation, transcription, and DNA replication. We go on to protein synthesis and so forth. um, the purpose of this lesson is to explain the basic introduction of DNA and the basic understanding of it. So as it is, I integrated an activity. Let's see, one strand of DNA has the base sequence of T A C G A T T G A. So when we talk about what's the complementary strand, as I spoke about the bases earlier, right? We talk about um, which base will pair with what. So what do we know? What what do we know? There we go. Thymine pairs with adenine. Adenine pairs with thymine. See, complementary bases. Cytosine says pairs with guanine. Guanine pairs with cytosine. Complementary bases will always pair with each other, even when you're doing activities. So the purpose of this presentation is to also engage learner interaction within the topic. Example also as well, when a learner is not is absent from school, they can still catch up onto the syllabus by watching the video or the presentations or slides uploaded by the teacher on the cloud or sent via WhatsApp or Google Drive, etc., where they can catch up with the lesson in a more simpler form as well. It can quizzes can be engaged into this slides where learners can engage and debate over it and take chances answering questions and understanding the module. Thank you.